All right, well, thank you very much, Asta, for that lovely welcome. And thank you to you and Katerina for the invitation to give the first seminar for the consortium. Welcome, everyone. I'm going to be talking about computer vision and its application in fisheries science. And I've put the word rapid in the title because I feel like we're on the crest of a wave where we've been working for some years on developing methods, and now we're applying it. And we're really something of a revolution in fisheries science. My opening image is fish and chips. Now, not everywhere in the world thinks this is a delightful meal, but there are some places, and there might be people, I know you're all in different time zones, so depending on the time of day, it might be sitting there making you feel a little bit hungry, that image. Others of you will think, well, that's not a good thing to do with a lovely piece of fish, is to put batter on it and deep fry it. But nevertheless, because we're talking AI, I'm not actually talking about those sorts of chips. I'm clearly going to be talking about computer chips. And before I move on to my next slide, I want to confirm really that this is the real Rod Connolly. I'm not a deep fake. Although actually, if I was a deep fake, I'd probably be programmed to say it exactly like that anyway. So it's up to you to decide. So I said I'd use some application studies. So these are 3 case studies that show how the computer vision can be applied. And I'm going to start with a relatively straightforward situation of asking about the success of mangrove restoration for fish habitat, and then move to something where the computer vision tasks are much more complex, much more demanding, about the recruitment of snapper onto reefs, and finally look at a straight fishery independent stock assessment situation and how computer vision is applied there. Now, I won't talk too much about the background of technique development, but well, firstly, let me introduce the team. The team's been broad over many years, but the current staff that have contributed to the work I'm talking about today are shown on the screen there, and I thank them very profusely. As the director of FishID, I've also worked closely with the GLOBE program, the Global Wetlands Project, and that's been co-led by Associate Professor Chris Brown. And then we have other colleagues at University of Sunshine Coast that we've worked very closely with for years and have supplied a lot of the videos that we've used as our ammunition to develop these techniques. Now, as a technique development, one of the big situations that people use is the baited underwater videos. We know that. I'm going to introduce the species here a little bit. This is a sparrowed. It's called snapper, Australasian snapper or pink snapper, but it's not in the snapper family. So internationally, this is a sparrow like the brims that a lot of people will know. This is a particularly large and long-lived one. And so it's important in my second case study, you'll see that actually biomass-wise, it's super important. And the work here was with David Fairclough and Gary Jackson at Western Australian Fisheries to work out how to get the right max N in an image like that, where the fish are clearly so prevalent that they're overlapping each other and there are bits of fish flying everywhere. And we managed to do that, and you can see the paper there in the bottom right. And while we were doing that, we also were able to develop the technique to look at time at which the fish first arrives from when the baited video goes in. So that's that point. And then throughout the video, we get the actual counts through time. So we can not just look at max M, but also look at mean count. So they're just a couple of touching on a couple of points that will come up later in the talk. So my first example is the restoration of mangroves as a habitat for fish. Not everyone will know this, so I'm going to say that there's a big groundswell in mangrove restoration. I mean, the Bezos Foundation is funding lots of on-ground works around the world, and that includes Australia, where I live. And that's because in many cases, the mangroves have been initially lost as land was reclaimed or the tide was prevented from getting to an area by new barriers and so forth. And part of or a very big part really of the reason for restoring is because they make good fish habitat. And the latest trend is to do environmental accounting on that. So if we're going to be spending money on the restoration, we want to know when we're going to get our money back. So the time lapse, the lag until we get fish using those mangroves is important to know. And around the world, it's not well known. And it's not well known because it's very difficult to sample fish in mangroves at all, certainly quantitatively. And so we've done that here, and the title might give away a little bit about what I'm going to show you we found. but it's more about how we went about it and got great data on exactly that. So in this case, the research objectives were to compare fish abundance between restored and natural mangroves at many locations, evaluate how the maturity of that site, how long since the mangroves were restored, how that affected the relative abundance of fish. And there was a third objective, which was to look at how fish utilise different zones within a mangrove forest. I'm actually not going to talk about the results of that today. I just wanted to leave it there because it shows the sort of nuanced questions one can ask with cameras and AI. And the suite of 4 species along the bottom are the ones that we focused on. We needed for this study, statistically speaking, to have species that were occurring widely across the different locations. And so these were the species that fitted the bill. The design was to have multiple sites. So we had 5 sites here spread along the coast of Queensland in Australia. And in each case, we had a paired design of restored and natural mangroves, the ones that had never been lost in the first place. And the orange number on the screen, on the map, shows how old the restored site is, from 1.5 years old through to 42 years old. And we use that like this when we analysed it. So this is time since restoration on the x-axis, and then this is the relative abundance. So this is the number of fish in the restored over the number of fish in the natural site paired. And a 0 here indicates that there's the same abundance in restored and natural. If it's above the line, then that means there were more fish in the restored than the natural, and below means there were fewer fish in restored. Theoretically, there are a few different ways that those fish might colonise the mangroves. Generally speaking, there were no fish to begin with because it was dry land, or the sea was unable to get there to that site. So it starts low, and then it could rapidly, the fish could rapidly colonise and match the abundances in the natural mangroves. Or that might be a more gradual process, or it might never happen. Maybe the fish will never match the abundances in the natural sites. So this is the sort of mangrove habitat we're talking about. And one of the striking things about it is that there's been very little quantitative data out of situations like this. And you can see why. There really isn't a type of netting or other normal practice for capturing fish, counting them and identifying them that can work in these sorts of habitats. I'm going to use cameras, you can probably guess, but before I get to that, I'll point out that it's not sheer laziness on my behalf. And I actually went and dug out an old photograph from my PhD where I'm in the background there, I'm at the back, and we're hauling a net full of fish in a beach seine net onto the shore to identify, count, and release. So I'm not afraid of hard work, but none of that is going to work in the mangroves. And so we go for cameras. And in this case, we put out banks of cameras along the fringe of the mangroves and also deeper into the forest. So those are the zones I was talking about. And then we set the cameras going, and as the tide comes in past the camera and the fish swim past the camera, we can identify and count the fish coming in. Now, Michaela, who led this work, we ended up with 500 hours worth of video. So you can imagine she didn't really want to sit down and do that manually. And she didn't. So she used the FishID processing pipeline and extracted the data automatically. So that's what it looks like. It's not pristine coral reefs, as you can see. It's more your murky mangrove water. So the data are extracted for those 4 key species. And the results for the first species, yellowfin brim, look like this. So we were looking for a pattern through time, and there is none. In other words, really time didn't matter. As soon as the mangroves are restored, the fish are using them in the same way that they're using adjacent mature natural forests straight away. Same for sea mullet and for toadfish. So there's no evidence of a trend over time. So that's an answer for the environmental accounting. It's for fisheries, for fish use, it's a very rapid use of those restored mangroves. So that's great new data. So that's the key finding. I'll end with one anomaly, which was the other species, which was the silver biddy. And this went completely the other way. In other words, They were very abundant, more abundant in the restored when there's hardly any vegetation there. And then as the vegetation matures, their numbers drop. And that's because they're a schooling species, somewhat pelagic, in a shallow sort of a way. And so open water, a bit more open water in the younger restored mangroves is actually favoured by them. Case study 2. Now, this is where I said we'll get more complex with our computer vision ask. This is a study of the way snapper and other fish, all other fish that occur as juveniles on reefs, are occupying these restored shellfish reefs. So this is across southern Australia. And in the top right, you see what it looks like from the air. Basically, we have unvegetated control habitat where we've lost the structured habitat over the last 100 to 150 years. And the Nature Conservancy and other groups are putting that habitat back. In this case, it's shellfish reefs. And you can see some of the images there from southern Australia. And what we're trying to do is work out what the fish biomass would be created or produced resulting from that restoration. And we're trying to do that quantitatively. First, let me show you what a bit of eye candy, what it might look like, and then I'll talk through the computer vision tasks. So we're putting in stereo rigs, and you'll notice the cameras are quite close together. And then there are fish at times teeming, not always. And we sample over the reef and also over unvegetated habitat alongside, such as that habitat. So lots of adult fish on those reefs, but not so interesting to us for this particular study. Yeah, they look good, and someone might want to catch them and eat them, or might want to enjoy the biodiversity of them. But those fish will have been potentially just living somewhere else and attracted to the reef, moved over to the reef. They're not necessarily short of adult habitat, so that's not necessarily new production. So we're more interested in this case in the juvenile fish, really the young of the year. So it's that settlement and initial survival that has been impeded by the lack of structured habitat, the removal of structured habitat. So in putting that back in, this is the size class that we want to quantify for each species. And we're going to compare the numbers of these fish on the reef against the unrestored, unstructured habitat. And we're doing that with the stereo camera. Now, a few things that we need to do, and I'm going to go through them sequentially. Well, we're going to need the size of these individual fish. And you'll notice that in this case, because we're after juveniles, we've used cameras that are stereo cameras that are quite close together, because we don't want to have that small patch in the middle right near. These are often going to be fish that are close to the cameras because they're very small and we want to be able to see them. And we don't want too big a gap between the cameras where you actually don't get them on both cameras. And to get the size and know whether they're a juvenile, we're going to need to know what species they are. We want to know species anyway, but the threshold at which a fish moves beyond its first year in size clearly varies among the species. And so we get the species, and then we're going to make counts, and I'm going to come back to what sort of counts we make. We also need the area over which that camera is surveying. So we need a density. We need it the same as if we were pulling a net, and we need to know the area over which that net was harvesting the fish. And to get the area, we get that from the stereo rig as well. So we know the distance that the camera is seeing, and then we know the geometry of the arc those cameras are projecting over. And from that, we get the area of seabed that is being surveyed. And I said that I'd mention count. So a lot of people know the count called max N, and that's not something we used here. Of course, we get the max N out of the videos, but it's not what we use because that would overestimate the density of fish. Because if we wait for an hour, for example, in that camera, and wait until there just happens to be quite a few fish here in front of the camera and say, oh, that's the density right at that moment, but that means those fish are not over in front of another survey area that might be being filmed or not. And so, long story short, we really are after the mean count through time. And so with the computer vision, we can get as many counts as we like through the videos and then take the average. And all of that's done using the FishID processing pipeline And I'm just going to show you a few seconds of what that looks like on these particular reefs. I said fish, but it includes the cephalopods as well, as you can see. And then we, apart from counting and identifying, we're sizing using the two streams. And that goes through to the size of each individual, as shown here. And that then gets tabulated, and I'm not going to go into details, but you get the idea, hopefully in the bottom right. This is an example of the density per fish per 100 square metres on the reef and unstructured. And then over on the far right is the difference column. And that's the one we're going to be working with from now on. That's the increase in fish density on the reef versus unstructured for these different species. This is actually just one example from one survey period from one location, because we need to do this over many times through the year, because the fish recruit at different times of year. So we then model using natural mortality rates and known growth rates for each species, and then summing that across the species here. We then estimate the total biomass, adult biomass, that would ensue from 1 year's cohort of young of the year, from 1 year's arrival of juveniles, living to adulthood with known mortality rates and growth rates. This is the productivity we would get in fish biomass. So if you're still with me on that, then over time, and remember, it takes a while for that cohort to grow up, because, for example, snapper can live for up to 40 years. Then look at the black line, which is the average across the locations, and we see that about 6,000 kilograms or 6 tons of fish biomass result from 1 hectare of restored reef. It's quite a substantial amount. We did all species, whether they were harvested or not, but largely it's harvested species that are contributing to the biomass, because typically they're the bigger animals as well. And then if I look at Australasian snapper versus all the other species, it's largely about snapper. They were there in great abundances as juveniles, and they're a large, long-lived species. So a lot of the biomass comes down to that species. And then finally, there were many other species involved as well. And so here are some of the top ones. If you want to go into the details, if you're interested in particular species, then that's clearly in the scientific paper that's supporting all of this. So what we have here is a really robust, reliable and repeatable survey strategy for working out, when we do this rather expensive restoration work of rebuilding shellfish reefs, what are we going to get out of it fish biomass wise? A big success of computer vision. My third case study is a classic stock assessment situation. We want fisheries independent data for, in this case, how many sea cucumbers are out there in the ocean. And so sea cucumber fishery around the world, whether they be small local fisheries, or in this case, large commercial fisheries with big boats, they worth quite a lot of money. For example, here for white teeth sea cucumber, you can set yourself back $300 to buy 500 grams worth, half a kilogram. And this is a collaboration seen in the bottom left with Macquarie University, the Great Barrier Reef Foundation, and the industry itself. And we're going to be using FishID to get us where we want to go in terms of doing this efficiently. So the industry and managers would like data from very many places, not just where the harvesting is done, but elsewhere, on the types of sea cucumbers we see there and the abundances. Now, that used to be done by divers who can do about a 200-metre transect, but now it's done with underwater drones, and they can go for a long time. If the battery lasts 8 hours, they can work all day filming like this, looking down on the seabed. You might have spotted a sea cucumber go past, and we'll see a few more in a moment. The point is that, as usual, it is a very, very efficient and effective way, much more effective and repeatable than having divers go in the water, and a lot cheaper as well, less expensive. However, it generates a huge amount of vision to look at. In this case, there are some challenges. For example, there are 3 species on the screen of sea cucumber. Only one of them is of interest or major interest commercially, and they look quite similar at first glance. That's one of the challenges. And the other one is simply the camouflage that some of these sea cucumbers achieve. So I'm going to play this very short video and let you see whether you can spot the sea cucumbers. There's a bit of sound to this, which is the sound of the drone as we move. All right, so you may have picked up a black sea cucumber there, depending on the quality of your screen. And there's another one that's doing a good impersonation of looking a bit like a rock, and it's in there. So we've been doing a lot of training of well-camouflaged sea cucumbers and even bits of sea cucumber that poke out from under a rock or a bit of coral. And that's all come together in a very effective computer vision task, which is to take videos like this and identify and count the sea cucumbers. I'll just let it play for a few more seconds so you get the idea. Sometimes there are none. We could go for a whole transect and see none, and then other times there are lots, and the counting is quite profuse like this. And then those data go to a database and are able to be accessed by whoever's uploaded the video, ultimately the fishing industry or the manager of the fishery, and they can get the data out, but they can also do QA, QC, quality assurance, and if there are any issues with identifications, they can go in and manually check. So that's the stock assessment example. Now, we actually work all over the world. We have many different examples. For example, we're counting the salmon swimming in the streams in Finland. All sorts of things. So I don't have time to go into them all today, but one particularly fun one was where the fisheries department in the state of Australia that I live in, Queensland, they've gone to a lot of trouble to put in artificial structures. These are not inexpensive structures. And they wanted to know how many individuals of the harvestable species occur there. And we use computer vision for that as well. And I have one more slide, but before I do, I want to pose a question or a comment really, and it's about how we talk to stakeholders in the industry about performance of our, in this case, computer vision or our AI more generally. And I have a smile about this because humans tend to think they are pretty reliable and usually get the right answer. I am a human. I said before I might be a deep fake, but I really am a human. And I'm not actually convinced, having seen the computer go head to head with humans so many times now. But generally speaking, the initial conversation is one of, well, whatever the humans are currently doing in terms of extracting data, we need to match that. And the closer we get to that answer, we consider to be doing well. But I parenthetically say, well, I have seen definitely instances where our computer vision algorithms are doing better than the human, not only more reliable, and whatever bias they have, it's the same every day, rather than depending on what day of the week it is and whether they've been having a big party the night before, as it happens to humans, but also actually better data and more reliable. Anyway, I'll pose that and we can come back to that in discussion time if people are interested. But I wanted to point, just last slide, I wanted to point to the future. And to do that, I'm going to go back a bit. So I call this fantastic beasts and where to find them. So this is the famous leafy sea dragon. And this is a signated fish. So this is a pregnant male. And it's endemic to Australia. And a long time ago, in fact, pretty much on 20 years ago, you'll see a couple of papers there where we were asked to track these fish around with ultrasonic pingers and determine their home range. And that was a very successful study that allowed the managers of the marine parks to design those marine parks to cover the home ranges of a certain number of sea dragons. But while we were doing it, we noticed that the facial markings allow us to recognize individual fish. Now, back then, 20 years ago, we took photographs, we manually drew those shapes, we had them on a dive slate, we would go down and we would recognize our fish. And I am really keen now to go back and do that properly using AI, now that we know that that sort of thing is just perfect for AI. And given that it was about 20 years ago today, I'm going to end with a small piece of music that captures that moment. So I hope you can hear it. That, thank you, and back to Asta as the host. Thank you.
Thank you, Rod. Yes, we could hear the music, and thanks for the very interesting presentation. It's well, I'm sure the audience has lots of questions. We can see claps in the audience. But it's great that you finished with this individual face identification, because I know in a few months we will have another presentation exactly about that, how we can identify individual fish, because there is a lot of progress in that as well, and so lots of applications. But now, so for security reasons, it was participants couldn't unmute, but we will enable all of this now, and you are welcome to ask questions. So maybe raise your hand first if you have some questions, and then you can unmute and start your video. So you can ask a question, and we left lots of time for discussions. But maybe I will ask the question first, since I'm here anyway. So I know you mentioned before we started that this is, we are now focusing on the application of computer vision, not how exactly to do it, but I know a lot of people here are really keen to know also how exactly to do it. So can I ask a question about that, even though you might want to ask questions to hear more questions about applications? But so the FishID, how many species can it currently identify? And is it available for others to use? Can I use it for my data analysis?
Well, firstly, how many species? So it's, as you know, the species vary around the world, and depending on the situation and the objective of the particular study. And so we train for that region or for that particular study. So we've got hundreds of species that we've trained it for, but whether that applies to your area, it depends on where you're thinking of working and what you're thinking of doing. So there's always a training component, well, nearly always, unless you happen to work with us, a training component for the particular target species that you want to quantify or count or size or whatever. And yes, so it's an open process where we do some training for anyone that wants to use it. And then after that, you should be away. Someone with your sort of expertise, with fishy expertise and a knowledge of AI, should be away on your own. A lot of the requests to use it are not from people like that. They're not researchers. They're people that really want to see the data, but they don't want to get their hands dirty with computing. And for them, we coach them all the way through, and we really provide a service for them.
Right. I do have a follow-up question, but I can see that Filippo has a raised hand.
Hi, Rod. Thank you very much for your talk. Can you hear me?
Yes, we can hear you.
That's amazing. Apologies for the video. My camera is not working, but your work is really inspiring. I have 3 questions about the FishID. So from what I understood, you have a model and then you fine-tune for different locations to work for different problems. What's the variability that you saw in these locations impacting the model? And then I'm also interested in how much training data you gather for these new locations to make the model be useful to the stakeholders. And then I'm going to go with my 3rd question. Thank you.
So yeah, thanks. They're all good questions, and they're on our minds all the time. So really good to be discussing this. So in terms of variability, I wasn't quite sure there whether you meant variability in performance or perhaps environmental conditions.
Environmental conditions.
Yeah, so they both come into it. So that's again, it depends on the particular situation. But you saw, if you saw the first example of working in the mangroves, where the water is really quite turbid much of the time, then we've done all sorts of procedures to make sure that we maximize the visibility. So by water clearing algorithms and so on, and then try to standardize that for distance from the camera. So that's a kind of a bespoke thing for mangrove and inshore work. But usually there's something to do with the environment that throws a spanner in the works, so to speak. Another good example is the need to train across habitat. So since we're still sitting on the screen with the gorgeous leafy sea dragon over the seagrass, if we train always over a background of seagrass, and then we end up also taking imagery over kelp-covered reefs and so forth, different habitats with different background, perhaps different dappled light playing on the rocks and so on, then performance can drop away. And so the idea is to train in a suitable way for what imagery you're actually going to be harvesting later when you do the project. Numbers totally varies depending. I mean, it can be really rather few instances or annotations needed sometimes for straightforward situations, and then using augmentation before we do the training. And but other times, if it's a tricky situation, like the camouflage with the sea cucumbers, then more training. So there's no specific number, but it can be from anything from small to lots. Happy to talk offline about that in more detail about your particular situation. And did you have a third one, did you say?
Yes. Just out of curiosity, based on your experience, do you see the field going more towards foundational models that generalize in different environments, or do you see it going more towards streamlining, fine-tuning for new locations?
That's a really good one. I mean, you've asked my opinion, so I'll give it, but I'm not to know how that's going to play out. But we're trying both, so we've got a foot in each camp, and I think that, well, put it this way: if we're in an aquaculture situation, which is more straightforward in the sense of at least we know where the fish are and we can manipulate where they will be in terms of relative to cameras or other sensors, then I think the bespoke, non-generalized models will probably win the day. And as I say, this is a wide confidence limits around any predictions I make here. Whereas for the wild fisheries and fish ecology situations, then it might be the more generalist models. Anyone else is willing to chime in on that? I don't have, I'm not the only one that will have a view on that one. Sebastian, I'll just introduce Sebastian Lopez Meccano, who's on the call. Did you want to, from the FishID team, Sebastian, if you're there and had a thought on what you've seen now and particularly what you think about the future on that issue, if you would like to comment.
No, I think I agree with you. But to add to the answer, I would say that the power of the foundational models will definitely make these fine-tuning or fine-tune models much quicker. So that obviously will increase the expectation of the humans on that, AI in underwater should work everywhere. And before we would take months to get a fine-tuned model working, but now with these foundational models we can get to the right answer much quicker. But yeah, the way that these, as you may know, the way that these foundational models are trained, usually for ecological applications, it's not sufficient. We want to have a species names or have it at a detail that it's useful for a statistical modeling. And if we're just labeling it as fish, then that's not really useful unless that's the question that you're looking for. So yeah.
Thank you so much.
Thank you. The next question is we have quite a few questions coming here also in the chat. So Katarina, I think you were next and the raised hand.
Hi, yeah, so I have a quick one. I also get some people ask me a lot about the reliability of the these AI models, and I was wondering do you think it's a case of the amount of data? Do you think with more images is it possible to always improve the model, and if the generative AI plays an important role in there, or do you think it's for some in some cases, it's not possible, even with good data set and a large amount of data.
Well, thanks, Katerina. Really good question. There was a bit in there to unpack, and I won't cover it all now, but just in brief. So more, like so often, then more training images helps, but not always. And then you need to use clever strategies. And so well, there are almost too many to go into now, but the general relationship is more data, but with single shot learning, zero shot learning, mixed models that use text and other information other than the image itself, which are all coming online now, then really I think we're in a pretty good position that you would think that there isn't too much that if you're, in my opinion, if your human eyes are, someone's human eyes are able to detect it, then the computer will be just fine doing it once we get the right training and models. Well, again, welcome anyone else's comments on that. It's a great question.
Rod, if it's possible, we could stop sharing the screen, then I think the audience will be better, will be easier to see everyone.
I enjoy doing that. I just thought if we had
and you can see people as well better. So the next question was from Gail.
Hi, thank you very much. It's been a fantastic session. Me as a mathematician rather than an environmental scientist, but Filippo's point was really interesting, raising the point that you use different training data for different situations. So really glad to hear that point. But I'm interested in what your current challenges are. This looks like an incredibly mature environment. But what are your current challenges? Are they mostly site-based issues, or is it technology? What are your technology challenges at the moment?
Yes, thanks, Gail. That's another big one. I don't actually see it as mature. I see it as very rapidly maturing technology rather than mature. So we're still learning lots along the way. Well, I mean, there are so many things I could say to that, but let me start with the finance side of this. So how do we fund the development of this? And it's to me like every automation situation that's ever existed. So when we were automating factories for making vehicles, cars years ago, then there's a big upfront investment, but in the out years, in the many years that follow, then the price of those cars or whatever they were manufacturing comes right down. And so it turns out for us that there's that hump in the need for funding at the beginning as we tailor our models and we do the training and so on, and we look for any inconsistencies or places of situations of poor performance and solve them. But once we do that, then the benefits are there for whoever's wanting the data. And so in a way, probably the challenge that I've seen is situations where someone is they have a budget that is the same every year. It's often the case for government and also for consultants that do a lot of work looking at fish and how many are in a particular habitat, for example. We bump into that a lot. And they don't have the funds to put in at the beginning and then reap the benefits later. Sometimes research grants are good for that, actually, because they do tend to work short term and then give you nothing down the track. But I think that's one of the challenges is that financial. And again, Sebastian, did you want to add any from your perspective? Is there a challenge on our doorstep that you're noticing a lot? So Sebastian is one of the scientists and also the project manager for FishID, which is why I bring him into the conversation like this.
Yeah, I would say that technical challenges, we don't have a lot. I mean, the way that all of these technologies, GPUs, models, and so on, making them available on GitHub and all that, it's definitely making it more accessible for everyone. I would say actually a challenge that is usually not discussed is, and I said it before, is the human expectation sometimes on the AI. We usually say if the human can see it, then the AI can do it. But what happens when you have an extremely rare, invasive, or super rare species where you cannot even collect any data, right? But then there's expectation that then the AI should do it. So we usually go through a training process of almost guiding people that there needs to be a limit sometimes on all these things. I hope that in the future we can take those really like important challenges for rare species, invasive species, when the training data is very null to minimal. But yeah, human expectation maybe is one of the challenges that is usually not considered. Yeah.
You. The next couple of questions are in the chat, and Rod, you can either read them yourself or I can read loudly. It's from Jeff from Adelaide Unidays. He's asking 3 questions. Do you track objects to ensure there is no multiple counting of individual fish? And how many images do you extract to train your classifiers for each species? So what's the size of the training dataset? Do you account for tracked fish with poor confidence scores? And how is your ultimate frisbee forehand going?
Let me start with the last one. I'm just as good at frisbee as I ever was. No, to the serious questions in there. So firstly, we do track objects. We don't always need to, but Sebastian's PhD originally was on the tracking and counting of fish. He's very good at it, and generally speaking, we need to make sure we don't double or triple count or get the wrong answer. Great question. How many images do we extract? So I think I mentioned before, that really varies a lot, from not many in simple situations to many thousands, more than 10,000, for example, in some situations. But we are using machine learning to extract to make that process of annotation very quick, as well as augmentation before we run the models. How am I going? Tracked fish with poor confidence scores. So we're playing around with the confidence all the time, depending on the situation. So sometimes where there's low confidence in say an identification or in the number of fish in a particular image, we are requested by whoever we're working with to tip that out for them to manually curate. So that's kind of like a basic thing that a lot of people like. It gives themselves confidence that they can look at the difficult images, if you like. But we also play around with the confidence thresholds a lot. And when I say play around, again, everyone on the call will probably know that if you like to lower your confidence threshold, then you're going to detect more of the species you're after, but you're also going to get more false positives, and vice versa if you raise the threshold. So that depends on the question and the situation. And thank you, Jeff.
Then there is a question from Jimmy Fries in the chat related to this, partly maybe not really answered about to the sea cucumbers yet. So how much training data and specifically annotations were required to ID the target sea cucumber? And also curious to hear about precision, recall numbers, or AP50 scores, or F1, whatever you like. So the technical aspect of that.
Sebastian, I'll hand over to you in a minute. That's someone that's so intimate in that project. But just before I do, I'll point out that this is almost going back to managing those expectations and what, in this case, you could almost call them a client. I mean, if it's the industry, then rather than a collaborator, although they are that to some extent as well, they're the technical aspects of performance, such as AP50 and recall and precision, are of interest to us, but in the end, what they want is the right number in a section, the right count of the right species in a section of video, which is a little bit of a different performance measure. And giving them numbers like these, which we'll talk about in a moment, because Sebastian's very good at them, and everyone on this call knows what they are, but the industry generally isn't interested. They're interested in how close you are to the right answer. Sebastian, do you want to just comment briefly on that question? About precision recall, for example, for the cucumbers.
Yeah. And just to complete the answer in a way. So yeah, we're more interested on the object counting, the same way that the human would do it when they see a section of video, rather than a particular frame and how much overlap the model's bounding box has on the human bounding box or annotation, right? Which is where all of this precision recall and F1. But overall speaking, for the counting method and also for more of the object detection side, the idea is to get at around 90% F1, and we're seeing that trend across the board. The number of annotations depend on how visible that sea cucumber is on the habitat. So again, it definitely does vary. One of the challenges that we have there that we're actively solving is, and I said it again before, sorry, that it's that some of the species are super rare, but they have a really large commercial value. So then how do you solve that problem of not having enough annotations or enough instances of video where you can actually extract that data? So that's when maybe other things outside of computer vision might come into play to solve that challenge.
Thank you. Freddie had a question.
Hello, thank you so much for the talk. My question was related to training. I was at a Fish AI symposium at AMSA, and it seemed like lots of people were working on similar species, but in different habitats. And someone asked a question about whether information from one system could be used in another system, whether they could be used to inform each other. Could that be the case with FishID? Can you use FishID to inform, say, one habitat for a species? Can that be used to inform a second habitat, or is that more confusing for the system?
Good question. I'm pausing only because there are so many angles to the answer to that. So let's go back a step. So for us that happens to use the FishID processing pipeline, it's that pipeline that's so efficient for us if we move to a new situation or a new question, and then retrain. So in a way, it's that both physical infrastructure or computing infrastructure and mental infrastructure, if you like, that human capacity to know how to do that is important. And then in terms of actual models and annotations, well, if the species are entirely different, like if we move really halfway across the world and are in a different pool of species of fish, then as far as I know, Sebastian might like to comment on this as well, probably those actual annotations won't help, but the model will end up being pretty much the same. So we're going to start with something of a generic model that we've already trained and get going pretty quickly. Again, Sebastian, would you like to just comment on that? It comes up, probably every week about how we're going to go in a different situation, in a different habitat.
Yeah, I think two things. The data sets and all the knowledge that you can, generate from one project, one habitat can be transferred to another project, another habitat, right? That can be done both from a human annotated data set, or it can be also done from a foundational model of what Filippo said at the start. So you can use all these old techniques to inform, to transfer the learning from one project to another. That's definitely can be done. A challenge there is that sometimes people are very private with their data sets. There's a lot of financial value on those data sets, right? And maybe something that as a group in this consortium might be good is to, promote open access of data sets. But yeah, so that's one thing. The other thing that will happen though, when you are, let's just say you have a model trained on one habitat and then are predicting or analyzing a new habitat, is that you almost get a shift, a domain shift, right? That you will see a decrease in performance. And that is a very well-known like machine learning AI problem. So our approach, and it's very standard across this industry, is that there always needs to be a little bit of new training data for that specific location. I have seen it personally in this fish space, in the coral space, where we were training different regions of the world. And then, usually because of how specific and, fine-tuned these models needs to be, there needs to be a bit of tweaking when you're working with the new habitats. But to get you started to get that fine-tuned model, definitely that transfer knowledge can definitely help.
Great. Do I have time, Asta, for a very quick
One
question?
more quick question, yes.
Yeah. So you mentioned about rare species, how it's the expectation of people with rare species might be too high. And then I thought about how humans would think about if I was going into a marine environment and I was looking for, say, a red handfish, and I hadn't seen a red handfish before, and I go to Google Images and I would look up images of a red handfish, and then I went down in person and I could see, okay, there is the red handfish. Is it possible to use still images for video analysis, or is that possible now? Is it possible in the future? Yeah.
Yes, it's
Rhoda.
Sebastian,
Yeah.
that was you that raised the rare species. Did you want to comment on that?
Yeah,
Yes.
yeah, yeah. No, sounds good. So yeah, the way that these most models are trained are with still images, right? It's just a frame that is coming from a video. So there's some temporal consistency there.
Yeah.
But if you look at the foundational models like Grounding Dino or Jono Nash or the World, all these things, they're trained all on still images from, let's just say Google, right? It could be anything. So yeah, there's definitely an opportunity there. I think where this industry is moving towards is that, you know, when you look at a rare species, you might use an identification book, and there's a description there saying that this species might be red, it has this scale and all these things.
Yeah.
Now with the new models where you can use image and also text, there's an opportunity there to also start adding descriptions to features of the fish that can help you without much training to identify a particular fish. But it's a very new, fast-paced area. Our initial testing have shown not too much success, but I would expect that will change in a week or 2 weeks or something, super quick. So yeah.
So exciting.
Week or 2 weeks, that's a pace at which you are working, right? So actually, I really want to pick up on the point about these data sets, of them being secretive, and that the whole community would benefit probably massively if we could share these data sets more effectively. That's one of the idea. So yes, let's try to work towards that. And had one more last question, perhaps, because we'll finish in a few minutes to let people get on with their days or go to sleep. So then, do you want to ask the question? Hi, then. All right. Oh yeah, there we go.
Hey, I'm sorry. Are you talking to me? So, thank you for this great talk. This was amazing, and there are a lot of things we need to follow up on. I'd like to ask you about your look ahead, and we've talked about individual ID. You know, so I'd like your thoughts about how an individual ID model could resolve some of the questions about, you know, fish biology in terms of schooling behavior versus competition, other things that come into play when it comes to, you know, max N. What are your thoughts about that? What are the training data, and what do you think when you look ahead towards that? Thanks.
Thanks, Stan, and that's a lovely question. If that's the last one, Asta, it's a good one to end on. We're lifting our eyes now and saying what's possible. And generally speaking, one of the things that I found with, let's say, fisheries scientists, but maybe it's more broadly the scientists there, although we're interested in innovation, we haven't actually been great to run ahead in our mind and think what's possible. So you'll find that scientists advising managers generally around the world are still using fairly tried and trusted and somewhat old-fashioned methods for gathering data. And that's not going to be how it is in the future. So when you think of a question about something, some very large area, it could be the whole of the Great Barrier Reef. How is it going? Or how is this habitat? Or how are the fish going in a big area across a whole river system, large river system? Then we're expecting to have lots of data, and I'm going to use an analogy shortly, lots of data from lots of places all of the time, and we're going to be more in a situation, and this doesn't specifically answer about the individual fish, Dan, but the way I see it, it's going to be more like the manager of a railway network who actually is sitting in front of a dashboard that shows whether every train is running and whether they're running on time, any delays, anything like that at all. And we haven't been used to that in environmental science and fisheries science, but it's coming to a dashboard near you. And as for individuals, I just raised that as one of the examples because I'm interested in it. And I think, Asta, you said we'll shortly have another, in sometime in the future, we'll have a seminar on that topic. Perhaps that'll be great. The questions are limitless, but certainly within aquaculture, we know that if we can measure the individuals without touching them, simply through the camera, and estimate their size, then we're clearly going to get great estimates of growth rates for individuals. So that's mean, but also variance around the mean, as one example. Thanks, Aston.
Yes, thank you. Actually, the upcoming seminar about individual identification will be from Tan, who is working on that. So it's great. We are looking forward to hearing more.
And very good. We were really looking forward to that. Thank you.
Yeah. So thanks a lot. And thanks everyone for joining. Thanks, Rod and Sebastian, for this amazing contribution. And as I wrote in the chat, the seminar recording will be available on our website. And there is also a discussion that we can continue on the website. And Sebastian promised to answer the questions. So make sure we use that opportunity. So more about links and resources and whatever other ideas. And we're looking forward to upcoming seminars and effective collaboration. Bye, everyone.
Thank you.
Thanks, everyone.